The overall goal of this procedure is to characterize the redox properties of an enzyme or protein. This is accomplished by first inserting a working and reference electrode connected to a voltmeter to a redox titration vessel above a magnetic stirrer. The second step is to add protein, buffer, and a mixture of different redox mediators to the titration vessel. Next, a chemical reductant and oxidant are used to poise the potential to different values, and electron paramagnetic resonance or EPR samples are taken. The final step is to record EPR spectra of the samples and prepare titration curves.
The main advantage of this technique over existing methods, like voltammetry, is that complex proteins with multiple different cofactors can be readily analyzed.
To begin this procedure, connect a silver chloride reference electrode and a platinum wire electrode to a redox titration vessel. Then, connect both electrodes to a voltmeter that can measure from millivolts up to volts. Insert a small stirring bar and place the titration vessel above a magnetic stirrer. Next, fill the titration vessel with previously prepared buffer B, mediator mix solution, and protein solution up to 2 milliliters to give final concentrations of 72 micromolar protein and 38 micromolar of each mediator. After stirring for 30 minutes, switch on the voltmeter. Introduce 10 clean quartz EPR tubes in an anaerobic chamber. Then